So, on Wednesday last week, we finished our conversation with the discussion about concentrations of the solutions. And for today, the only couple of things that I wanted to chat about: osmosis, okay, dialysis, the biological applications. So basically, the definition of the osmosis is the flow of water from the area with a lower concentration of the solute to the area with a higher concentration of the solute. Okay. Basically, osmosis is an example of water diffusion. So if you would look at this beaker right here, okay, you see that on the left side of that beaker, there are no solutes. And on the right side, there are some solutes. That makes sense? So right side of the beaker is composed of two types of molecules. Molecules of water and molecules of solids. Does that make sense? Mm-hmm. Now I'm going to ask you an unconventional question. What is the concentration of water on the left side of the beaker? Mass percent, volume percent, doesn't matter. 100%. Does that make sense? On the left side, right here, it's only water. So that's it. It's 100%. Now, the concentration of water on the right side of the beaker, it's not 100%. It's less than 100%. Because some molecules are water molecules, and some molecules of solute. So water does not comprise 100% of all molecules. Does that make sense? Mm-hmm. Meaning that water concentration isn't 100. Mm-hmm. It's less than that. Which means that the water concentration on the left is higher than the water concentration on the right. And then, just think about it. Diffusion is essentially a movement of any molecules from their high concentration to their low concentration. Does that make sense? However, in real life, we never operate with the terminology of water concentration. We do not express the concentration of solutes as water concentration. Does that make sense? So we say water goes from low concentration of solutes, there's more water, to high concentration of solutes, there's less water. Make sense? Okay. Now, if you will do the mental experiment, or real experiment, doesn't matter, like the one that's shown here. Okay? And you will separate this beaker into halves with a semi-permeable membrane. Now, why semi-permeable membrane? Because this membrane must be permeable to the water molecules, but not to the ions or glucose or something else. Does that make sense to you? This is why we call it semi-permeable. So, as water will flow from the left to the right side of the beaker, what's going to happen to the level of the solution at the right side of the beaker? It's going to become higher. Now, how does the pressure of fluid changes as its level increases? Yes, pressure becomes higher. Does that make sense? The higher level of fluid, any fluid, you can take water, solution, gas, doesn't matter. The higher level of the fluid physically will exert more pressure. Does that make sense? So at some point, the flow of water that is caused by osmotic force, so that flow that I showed here in that red arrow, will be compensated by the flow of water stimulated, sort of caused by that pressure difference. Does it make sense? So you're going to have some water molecules going from left to the right side of the beaker because of the concentration difference, correct? And then there will be pressure kind of pushing that water back. Are we clear? At some point. Does it make sense? We're good? You sure?
Yeah.
So basically, okay, let me picture. So in the very beginning, this is what you have. So those are levels of the fluid, and that's in the middle. Here is semi-permeable membrane. Does that make sense? Now, after a while, what you're going to have is the lower level on the left and higher on the right. And it's not going to, on the right side, it's not going to go up all the way. At some point, it will stop when the pressure, just hydrostatic pressure, pressure of the fluid, will compensate for the osmotic flow. That makes sense?
So the osmotic pressure on the right-hand side is higher.
Osmotic pressure is only on the right-hand side. And that osmotic pressure compensates for the flow. Does that make sense? So, osmotic pressure is basically equal to the pressure that will prevent that flow. That's how we can calculate it, right? That makes sense? So, and there's another important consequence of it. So, when you will take two solutions, two beakers, okay, let me erase it and picture two beakers. If you will have two beakers, okay, and this is pure water, so the concentration of solutes is 0, and on the right hand side there are solutions with the concentration C1 and C2. If C2 is bigger than C1, then the osmotic pressure in that system on the right hand side will be higher. Does that make sense? Now, how is that important? In physiology and in infectious diseases. Let's start with the human physiology. So it's going to be sort of a crash course, okay? You have blood in your blood vessels, right? Eventually this blood reaches tiniest, smallest blood vessels in your tissues that are called capillaries. So far, you with me? And the fluid in your capillaries and in your tissues, the fluid is driven by several pressures. First of all, it's driven by the blood pressure. Okay, so blood pressure drives the fluid through the capillaries, and essentially, blood pressure drives the fluid out of the capillary. Does that make sense to you? Is that clear? Okay, it drives the fluid out of the capillary. But on top of that, you have proteins, colloid, in your plasma. So far, so good. However, you have proteins in the plasma, but not in the fluid that is found in your tissues outside of blood vessels. So basically, you have a system where you have a blood vessel, and proteins are present in the blood vessel, but not in the tissue outside. So concentration of proteins is higher in the blood than in the tissue. Are you following me? So concentration of a protein is higher in the blood, which means another flow will go from tissues back in the blood. So you have, generally speaking, two main flows. One, hydrostatic, your blood pressure drives the fluid out of the capillaries. Does that make sense? Good. Another, osmotic, drives the fluid back into the capillary. You with me so far? In the perfect situation, in a healthy person, these flows are kind of matched. They aren't, but that's not the point. Now, what if, so you have a system, let me erase it and show you what I'm talking about and why osmosis, understanding of osmosis is so important in physiology. So let me draw it again. So I have a blood vessel, okay? Okay, that's blood here. And you have blood pressure driving the fluid out. And you have osmotic pressure driving the fluid in. You with me so far? Imagine somebody experiences grossly increased blood pressure. It means that more and more fluid will be driven out of the blood into the tissue. Does that make sense? And if fluid goes into the tissue, what you going to have? No. What are you going to have? Edema. Okay, if it's your, like, for instance, if you stand for a long time, if your work is, standing for a long time, you often experience, swollen feet. That's edema because of the elevated pressure because you're staying. That's the gravity. Okay, does that make sense? Now, if that elevated blood pressure starts to drive fluid from the capillaries in your brain, you're screwed. That's the brain edema, which may eventually lead to death. Makes sense, right? Now, what if patient's liver starts to fail? Well, it turns out liver is responsible for producing plasma proteins. When liver fails, concentration of plasma proteins goes down. When concentration of plasma proteins goes down, how does it affect that osmotic flow? Think about this. Fluid goes towards higher concentration. If the concentration becomes less and less higher, how will it affect the flow? Increase or decrease? Huh? So fluid goes to the higher concentration. That higher concentration, due to the illness, becomes lower and lower and lower. Osmosis will not be as effective. Does that make sense? Remember our experiment with osmotic bags? The biggest change in the weight of the bag we observed when 60% dextrose was placed in the water. Does that make sense? So if you have an anomaly, if you have liver failure or liver disease, and your liver fails to produce sufficient amount of blood proteins, the fluid will stop going from your tissues and will remain in your tissues. And what you're going to develop? Again, edema. Okay? One of the signs of the liver failure. Does that make sense? So that's kind of pathological applications of osmosis. Another important application of osmosis is absorption of various ions in your intestine. So without going into the details, but the first ion that is absorbed in your intestine is sodium. Okay? Sodium and chloride, salt, right? So as salt gets absorbed into your tissues from your intestine, how does it change the concentration of salt in the tissues? If it's absorbed into the tissues, its concentration in the tissues goes up, correct? Now, osmosis is the flow of water towards the higher concentration of solutes. Does that make sense? If a solute concentration, like concentration of sodium chloride, in your tissue increases, are you following me?
Yes.
Water from your intestine will be absorbed in the tissues. That's how you absorb water. That's the normal physiological mechanism. Now, if for some reason you fail to absorb sodium chloride and it stays in your gut, water will stay in your gut too. And if water stays in your gut, what symptom or sign are you going to experience? Exactly. You're going to experience diarrhea. Does that make sense? Finally, the last bit about osmosis. Does anyone remember those golden days when KFC produced a wonderful sandwich where they replaced 2 buns with 2 pieces of fried chicken? This sandwich, one sandwich, contained twice as much sodium as a person should consume in a day. That single sandwich. Now, imagine what happens when you eat that enormous amount of salt. Eventually, all that salt ends up in your blood. You with me? So, concentration of salt in your blood goes up. How does it influence the osmosis of water into your blood? Increases, exactly. Does that make sense? So water starts to rush into the blood due to the osmotic forces. When water rushes into your blood, what happens to your blood volume? It increases. But remember, all these pipes that we call blood vessels, they have sort of limited capacity. So when your blood volume increases, the pressure in the system increases as well. I spoke to a guy who actually did that inhumane experiment and ate the entire thing. And he said he felt it. Like his blood pressure spiked up. He didn't have to measure it. It was like physiologically, he would feel it. That makes sense? So I can tell you that understanding the process of osmosis, when you get to some extent AP1, but particularly AP2, digestive system, renal regulation, cardiovascular regulation, it will be priceless for you to know that and understand that, and it will give you a huge head start over other people. Okay? Reverse osmosis. So you have 2 solutions. A very diluted one, well, basically distilled water, and the water that you need to... Okay, let me start over. You have a membrane, okay? You have concentrated solution on that side. Okay, concentrated solution means it contains water and solids. You apply pressure, physical pressure, on that concentrated solution. And the pressure is so great that it pushes water molecule through the membrane. Does that make sense? The membrane is designed in a way that only water can cross it. Make sense? When the water appears on that side, of course, that immediately creates an osmotic pressure that drives water in the opposite direction. But your physical pressure here is much greater. So you keep pushing water out. Reverse osmosis devices are used to produce deionized water. Water that does not contain any solutes whatsoever. Okay? It is ridiculously expensive because it requires an enormous amount of energy. But, and generally speaking, you know, countries like the United States, Canada, European countries, the only applications that we need reverse osmosis for are lab applications. Does that make sense? So basically, you may have a device in the lab that produces highly purified water at decent rate. I don't know, like you can basically get some fresh deionized water, half a liter, or something like that. But can we use it to desalinate seawater and produce fresh water? The answer is yes. Again, it is ridiculously expensive. And guess which countries use reverse osmosis to produce drinking water? the Persian Gulf countries. Countries that produce the vast amount, the most of the world's oil. They spend a bit of their wealth on actual, like they have desalination plants that produce fresh water for utility usage. Kuwait, for instance, spent a lot of money on building desalination plants at the Red Sea to produce water because they have no other fresh water sources whatsoever. Does that make sense? Now, something that we covered in the lab a little bit, just wanted to kind of go over it a little again. So isotonic versus hypertonic versus isotonic solution. And we're going to come back to the idea of osmosis and movement of water in regards to the microbiology. So isotonic solution. Solution which exerts the same osmotic pressure as the bodily fluids do. Okay? Now, one important thing about osmotic pressure. Osmotic pressure depends on the concentration of particles, but it doesn't really depend on the nature of particles. Does that make sense to you? Let me explain. So here, example of isotonic solution is 0.9 mass volume percent solution of sodium chloride. That is typical IV fluid, isotonic solution. Does that make sense? Now, what if, hypothetically, you replace sodium chloride, like hypothetically, just for the lab purposes, with potassium chloride? Does that solution be also isotonic? The answer is yes. Because the number of particles in the solution, 0.9% sodium chloride, and solution that has the same amount of particles, same moles of potassium chloride, will have the same number of particles. Am I clear? It is all about the particles. It's not about the nature. You see what I'm saying? Is that understood? That's really important. We don't use potassium chloride for different reason, because if you give a person a lot of potassium in the blood, person will develop ventricular fibrillation and die. That's, by the way, how capital punishment is carried out in the United States. Injection of highly concentrated solution of potassium chloride along with painkillers and sleep medicines. Okay, but potassium is, in high concentrations, is toxic. But nevertheless, the idea that I try to explain here is that it's all about the particles and how many of the particles you have in the solution, but not which particles you have. Does that make sense? So, isotonic solution. In isotonic solution, cells do not change their volume at all. We clear? However, if you put cells in a hypertonic solution, they will start swelling. Okay. Don't change anything. Don't change volume. Okay. Don't change size. All right. Now, in hypertonic solution, what you get is, let's say, you have a bag which contains 0.9% sodium chloride, right? And on the outside of that bag, you have distilled water, which means 0%. So which solution is more concentrated? The one in the bag. So water will move towards the higher concentration across the semi-permeable membrane into the bag. Now, in our experiments that we did in the lab, when we used the dialysis tubing essentially represented the cell membrane. Does that make sense to you? That was the cell membrane, semi-permeable membrane. Okay. And yes, cell membrane is selectively permeable for water, ions, and other small molecules. When water was flowing into our bags in experiment, they increased in size. They got swollen, right? So there are few implications in regards to life science. In human physiology, if you will try to inject distilled water into a person, into bloodstream, the person will experience the massive hemolysis. Okay, red blood cells will start swelling and start popping. Okay, actually, I mean, you need a microscope for it, but if you bleed yourself and you add a couple drops of blood to distilled water, if you wait long enough, you can see red blood cells popping up. You can see hemolysis under the microscope. Okay? So that's one implication. Another implication is for microbes. Microbes often live in very diluted environments, like, fresh water, like lakes and rivers. So, for instance, bacteria are protected from bursting by the cell wall. Now, going back to humans, because bacteria start to swell, that's normal, but cell wall doesn't let them explode. Going back to humans, have you heard about the competition that happened quite a few years ago in California? They had a competition, who will drink more water. A woman won, drinking 6 gallons of water. She died next day. In her home, reason for it was, imagine what happens to the concentration of all solutes in your blood and the fluid that surrounds your cells after you drink 6 gallons of water. I cannot even imagine. Your kidneys cannot get rid of that much water. They're physiologically incapable to do that. Does that make sense? So that water basically accumulates, dilutes your blood, and your cells that used to be in the normal isotonic environment start to be in hypertonic environment. As they are in the hypertonic environment, they start to swell. Okay? And, I mean, it's not good, obviously, but the worst part, your neurons start to swell. Cells that are in your brain. Okay? And as they swell, they stop functioning. Person died. I don't know if they sued the radio station or not. But it's kind of what happens. You do stupid shit. Hypertonic solution. Solution with very high salinity, very high concentration of solids. Okay? So in hypertonic solution, the cell will shrivel because if you have, say, 0.9%, on the outside you have 10%, the water will flow towards the higher concentration. The volume of the cell will increase. Oh, it will decrease, I'm sorry. Cell will shrivel. It's called crenation. Does that make sense? So that's how we protect our food. We make the environment around our food extremely hypertonic for bacteria and other microorganisms. We salt our food. Salt protects it. Works really lovely. Does that make sense?
So the cells in hypertonic solutions shrink?
In hypo or hyper?
Hyper.
Hyper, they become shriveled. They decrease in size.
Okay.
Like you see here, red blood cells,
Uh-huh.
the spiky little things, that's how they shrivel. Okay. And you can actually see that too if you use, say, 5% sodium chloride, and 5 mass percent sodium chloride, and you drop red blood cells, you drop blood in there and observe red blood cells, they will be spiky, noticeably spiky on the regular microscope. That makes sense? Now, one of the applications of semi-permeable membranes is the process called dialysis. So I'm familiar with dialysis in the lab applications. Okay? So, basically the idea is that the membrane of a dialysis bag is permeable for water and small solid particles. By small solid particles, we mean like glucose, sodium chloride, like different salts, but that's it. No protein, no starch can cross. Okay, large carbohydrates cannot, proteins cannot. So let's say I work in the lab, I work on some project, and I have isolated a protein. Okay, during protein isolation, usually you use a lot of various salts, so you end up with a protein preparation that contains a lot of salt, which makes it useless if you want to experiment with it, because salt will interfere with a bunch of chemical reactions. So you take your preparation, you put it in the dialysis bag, okay, and you basically throw it in the water. And over time, water will move into the dialysis bag. Does that make sense? And it will start swelling, which is like hilarious, because you drop a tiny little bag, you come next day, the bag is ginormous, okay? And glucose, salts, whatever you have in your protein prep will move out, will go into the solution in the environment, okay? So now you have your protein in a pretty decent environment. You can work with it at least. That makes sense. Now, dialysis is used medically in patients with failing kidneys, yes. So your kidney is the organ that filters stuff out. Okay, it's not liver. No. Liver doesn't filter things out. It's kidney. Okay. Kidney works in a very interesting manner. I compare it to a person who, like, after, that Marie Kondo with cleaning closets on Netflix, I just saw it, decided it's completely, like, stupid to do that. Who am I to judge? Anyway, so basically kidney cleans up the closet, which is your blood, in a very interesting way. First takes everything and throws everything away, just everything. Everything except water. Like everything goes out. Even water goes out. Like everything goes out from your blood. And then kidney looks at it and says, well, I kind of need 99% of what I just threw out. So 99% of what was filtered goes back in the blood. But some stuff doesn't. And that stuff that doesn't eventually leaves your body with urine. Okay? And perhaps the most important, a lot of molecules living and ions living in your body with urine, some excess of calcium maybe, some potassium will leave definitely. Most importantly, it's going to be urea. Okay? Does that make sense? So urea is the product of the protein breakdown in your cells. If anyone ever tried to go on extremely high protein diet, you will start producing a lot more urea than the average person. Your urine will have bright yellow color and will smell funny. It's totally normal, but it's going to smell funny because of the urea and ammonia. Okay? And your kidneys will filter it out. It's fine. But if a person's kidneys fail, urea doesn't get cleared out of the blood by kidneys, and something else needs to be done. So that's why those people go to the dialysis machines, where combination of solutions outside of artificial dialysis membrane will basically force urea to diffuse through that semi-permeable membrane from the blood into the surrounding area. Does that make sense? And actually, that is one of the symptoms of kidney failure, elevated levels of urea in the blood. One of the symptoms of kidney failure, elevated levels of urea in the blood. Make sense? Any questions? Nope? Okay. So we're going to